you, Inês. Good morning, everyone. We will continue with our participants communication session. Will present us the main results of their research in developing and applying natural products in the health area. Let's start with the pitch sessions. I want to remind the audience that you can follow the conference's live streaming on YouTube's IPB channel. You can follow the communications and you can send your own questions through the chat. And after selection, I will present those questions to our speakers. You can also download the posters available online. You will find on the conference website 3 links corresponding to the 3 areas covered in this Congress: health, food, and cosmetics. Regarding the pitch sessions, I inform our speakers that they have 3 minutes to make their presentations. There will be no interruption between the presentations, and only at the end we will open a space for questions. To not cause major delays for this pitch session, we will allow you to ask only 1 or 2 questions. The remaining questions could be sent by email, okay? So let's move on. Ana Sofia Pereira de Freitas will start the session. Ana, you can start your presentation. You have the floor.
So, good morning, everyone. My name is Ana. I'm a PhD student from the University of Minho, Portugal, and I will present a part of my work regarding the antibacterial and antioxidant synergism between propolis and gentamicin or honey. Since the first case of bacterial infection with resistance in 2008, antimicrobial resistance has become an emergent problem, causing a prediction of 10 million deaths globally by 2050. The overuse of antibiotics, their extensive use in farming practices, their patients' misuse, along with the lack of new antibiotics, are contributing factors to this problem. So there is a need for strategies to overcome the microbial resistance and development of new antimicrobial drugs. Interest in natural products has been increasing due to their spectrum of bioactivities that can be used for the development of new drugs, to retard the outbreak of bacterial resistance, to decrease bacterial resistance impacts. So propolis is a natural product produced by bees from several plant materials and mixed with their salivary enzymes. So after the determination of the minimum inhibitory concentrations for both gentamicin and the ethanol extracts of propolis, by the agar dilution method, several mixtures of submic concentrations of both agents were tested in order to find synergism. So as you can see in this table, with a submic of 25 micrograms per milliliter of propolis, it was possible to reduce to 25 times less the amount of gentamicin initially needed to inhibit the growth of bacillus strains. Also for Staphylococcus aureus methicillin sensitive and resistant strains and for E. coli, it was possible a reduction of 70% of the amount of gentamicin initially needed to inhibit bacterial growth. Regarding propolis and honey synergism, the higher the percentage of honey in the mixture, the lower the antioxidant capacity. However, with the exception for Bacillus cereus, all the mixtures of submic concentrations of propolis and honey showed strong antibacterial activity than each individual sample, with particular interest for these strains, where it was found a huge reduction of the amount of both agents. So this outcome is significant considering the worldwide problem of antibiotic resistance and the need to innovate in antibiotic therapy, in particular if solutions from natural sources, such as propolis and honey, are envisaged. So thank you for your attention.
Thank you, Ana. Thank you so much. Now we are moving on to André. André Folgado, you may present your poster, your pitch session, please.
Yes, so you are hearing me? Okay,
Yeah.
So thank you. Good morning, my name is André Folgado, and today I'm going to talk to you about area root cultures of Cinera cardunculus as a valuable source of hydroxycinnamic acid compounds. So, as you already heard, cardoon is a plant widely studied regarding their secondary metabolites. The upper parts of the plant already showed some important secondary metabolites. Although this is true for the above ground part of the plant, the underground part of the plant still remain pretty much unstudied. To address this question and this issue, we used area root cultures. So these area root cultures are the result of Agrobacterium rhizogenes infection. These are characterized by a high growth rate and also high branching. They are important because they produce the same secondary metabolites as the source plant, and they are characterized by a high genetic stability, which give this a high potential for biotechnological applications. So for the induction of the air roots from cardoon, we infected hypocotyls from cardoon, and after we had the air roots, we perform air root methanolic extract, and we analyze this extract by HPLC-MS, and we detect several compounds, although this mixture showed low complexity. So the peaks are very well resolved. So 3 of them were confirmed by standard, and the rest of the peaks were confirmed by comparison with the literature. All of these compounds belong to the hydroxycinnamic acid group, and all of them are chlorogenic acid derivatives. Then we analyzed these cultures in their growth curve, and we analyze them in terms of total phenolic content that increases along the growth curve of the air root cultures, and the same pattern is true for the antioxidant activity, reaching its highest value at the 35th day of the growth curve. So we use this a time point to produce a concentrated extract that was further used to test antiproliferative activity on human colorectal cell lines, HT29 cells, that presented an EC50 at 1.16 milligrams per milliliter. The same concentration showed no effect on cells that mimic the intestinal epithelium cells, human cells. So with this, I thank you, and I finish. Thank you for your attention.
Thank you, André. Now, Bruna Mara Machado Ribeiro will present her pitch. Bruna?
Good morning. Okay?
Okay.
My name is Bruna Mara Machado Ribeiro. I'm from Brazil, work as scientific research. My line is neuropharmacology, neuroinflammation, neuroprotection, and neuroimmune activation. My studies detect inflammatory effect of N3 polysaccharide fatty acids on virus infect neurons. The viral mimetic poly-IC is important to study the consequences of viral infection to the development of neuropsychiatric disorders. Based on the premise of omega-3 presents anti-inflammatory effect, was investigated the involvement of NF-kappa-B pathway in the effects of omega-3 in hippocampal neurons exposed to the polycell virus. Poly-IC is a synthetic immunostimulation virus. Poly-IC induced by microglial activation. Poly-IC increased to levels in NOS, NF-kappa-B, P5, P65, ELIC, nitrites, and hippocampal neuronal cells. Poly-IC causes deficits in neuronal hippocampal mechanism, Ômega 3 previne neuroinflammation alterations induzidas por bile-poly-IC. Ômega 3 previne the deficits de BDNF. Ômega 3 increases DCX immune expression. Ômega 3 presents effect anti-inflammatory. Poly-C induced microglia activation. Poly-C induced increased the levels of NF-kappa-B P15, NF-kappa-B P65, ômega 3 effect anti-neuroinflammatory, effects neuroprotectives ômega 3 in neuronal cells, MTT increased, neuroprotective effects ômega 3 lipid peroxidation, ômega 3 suppresses the expression of P15, 65, NF-kappa-B subunit, neuroprotective effects of ômega 3 in NOS, neuroprotective effects ômega 3 in P15, NF-kappa-B in NOS, co-immunostent, in primary hippocamponeuros, neuroprotective effect ômega 3 in P65, NF-kappa-B, protein expression in primary hippocampal neurons, changeling, neuroprotective effects ômega 3 in interleucina 6 levels in primary hippocampal neurotendons, poly-C, neuroprotective effect of ômega 3 in TREACH levels in primary hippocampal neurons, ômega 3 previne the deficits BDNF in cells, ômega 3 increases the CGX immunoexpression. Thank you very much. Good morning.
Thank you, Bruna. Now I call Catarina Faria Silva to make her presentation. Good morning, Catarina.
I will share my here. Okay, can you hear me?
Yeah,
okay.
Perfectly.
So good morning, I'm Catarina Silva from Faculty of Pharmacy of the University of Lisbon. And today I will tell you about the extraction of alpha-tomatine from green tomatoes industrial waste to treat skin inflammatory diseases. Starting with the green tomatoes. During the tomatoes harvest, many of them are still green and they are thrown back to the fields, being an industrial waste. Furthermore, it is known that the green tomatoes are rich in glycoalkaloids, namely alpha-tomatine. And this glycoalkaloid was already studied in its pure form by several research groups, and they found out that it possessed several activities, namely anti-inflammatory, fungicide, anti-carcinogenic activity. So what we are proposing here is to establish a sustainable and circular economy where we are going to take the industrial waste of green tomatoes, extract the alpha-tomatine through an environmentally friendly method, the subcritical water, and then we are going to incorporate the alpha-tomatine in a topical formulation to treat the skin inflammatory diseases. The extraction, this thermodynamical process is a very important part of this work because we want to assure that the process is as green and sustainable as possible. And subcritical water basically uses only water, high pressure, and the tomatoes. We had to develop these methods. We tested several conditions, and in the end, we obtained an extract. To have a comparison, we also performed the extraction with conventional organic solvents with a method that is already described in the literature. However, the extract has all the components of the tomatoes. So in order to quantify the alpha-tomatine, we need to do an additional purification step. So we perform solid phase extraction, and what we found out is that when using the subcritical water extraction, we obtain 10 times more alpha-tomatine than with conventional organic solvents. So what we are going to do next now is to do a characterization of the full composition of the extract, and thank you for your attention.
Thank you, Catarina. Now we have Hala Haddad.
Hello.
Hello, good morning. You can make your presentation.
Yeah. Hello. Hello, my name is Hala, and today I'm going to present a project titled by Optimization of New Anti-Inflammatory Methodologies to Evaluate Natural Product Activity as Inflammation Inhibitors. I'm going to start with the introduction and objectives. Inflammation is a defense mechanism that is designed to eradicate microbes or irritants to protect living tissue from infections, injuries, and to potentiate tissue repair. The objective is to exploit Zingiber officinalis and Bruna dioica as promising anti-inflammatory agents. Methodology. We are going to use the hydroethanolic extract to evaluate the anti-inflammatory activity using mouse macrophage, cytotoxic activity using 4 tumoral cell lines and 1 normal cell lines, and the cell cycle and apoptosis using flow cytometry. Result and discussion. The results are still preliminary and needs to be repeated. The anti-inflammatory activity. We are going to measure the EC50 value, which is correspond to the extract concentration achieving 50% of the inhibition of NO production. The two samples and their combination present a really interesting value, and the BD present a very low value, which is even lower than the positive control. Cytotoxic activity. We are going to measure the GE50 value, which is correspond to the concentration of extract that inhibited 50% of cell growth. For Z officinalis, the 4 cell lines present a low GE50, a lower GE50 than the normal cell line, and the NCE present the lowest one. For BD, all the value of GE was lower than 6.25, so we are going to repeat this test with the lower concentration. The cell cycle analysis shows that the Z officinalis had the same cell distribution as the blind test, and the B dioica has a really interesting increase in G0 and G1 phases, so it does interfere with the cell cycle. The apoptosis analysis shows that the BD have no apoptotic effect, and that the ginger, which causes a really interesting decreased percentage of dead cells, have an apoptotic effect, which is confirmed to the literature. Conclusions. Given the well-known side effects of the commonly used synthetic anti-inflammatory agents, this preliminary study contributes to the discovery of natural agents as real alternatives and with less or no side effects. Bruna dioica revealed the highest potential with the highest anti-inflammatory activity and the higher effect on the cell cycle distribution. The chemical characterization in terms of phenolic compound profile is ongoing, and that's really interesting parameters, so we can know the relation between phenolic compound composition and the biologic activity. Thanks for your interest.
Thank you, Hala. Finally, I invite Naiara Fernández to present her work. Naiara, good morning. Is Naiara there? Yeah. We are not listening to you. You have your microphone.
I'm really sorry. I'm already sharing my screen?
Yeah.
Okay, perfect.
Okay.
Thank you. Sorry for the delay. Okay, so I'm Naiara Fernández. I work in Nutraceuticals and Bioactive Process Technology Lab at IBET. And today I would like to show you a little bit about the industrial waste stream valorization that we are doing here at IBET. The competences in our food and health division includes the recovery of high added value compounds, chemical and bioactivity characterization, and finally, if needed, we also do some formulation for the addition of these ingredients in final products. We are used to work with different natural matrices, but today I will present 3 case studies that are based on wineries and fisheries waste streams. In this first case study, I would like to show you several terpene-based natural dipotectic systems were synthesized with the aim of recovering astaxanthin from brown shell residues. So those systems were synthesized and physicochemically characterized, and they're applied for the destruction of the mentioned carotenoid. The results show that the results were comparable to the ones obtained with conventional methodologies. And also they were applied to another matrices such as microalgae and other seafood. Also, this terpene-based system and the extract obtained showed to have a good potential for anti-cancer and antimicrobial activity. This second work that I would like to present to you, we were able to extract phenolic and anthocyanin compounds from winery waste streams. We were able to enhance the yield of the extraction, applying a microwave pretreatment prior to the solid liquid extraction. And then those extracts were evaluated for their potential for the possible potential in cosmetic applications. They saw high antioxidant activity, both in chemical and cell-based assays, and also several enzymatic assays showed that they had some effect in skin aging. Finally, I would like to show you this cotton-like extractor, a fiber that was synthesized here by supercritical drying of gel that was formed by alginate and chitosan, both of them very abundant polymers in marine environments. So this material showed to be non-cytotoxic and also showed to have a strong antimicrobial effect. Moreover, it stimulates the migration of skin cells. So overall, it was seen like a possible candidate to use in wound healing applications. And that's all. Thank you for your attention. Thank you for the conference committee for allowing me doing this presentation. And please, as it was very fast, if you have any doubt or comment, please contact me by email and we can discuss further. Thank you.
Thank you so much, Naiara. We have here one question. First of all, thank you all that present your pitch. All were very clear. And I have here a question sent by Marcio, one of our participants. He thanks Catarina for her presentation. And he thinks it's a very nice idea. So Catarina, Marcio is asking, why did you choose the subcritical water to perform your work? Why? And if you choose another one or tested another one?
Well, like I said, we compared with the organic solvents that is already used in describing the literature, but the subcritical water allows us to have what I show much more tomatine, alpha-tomatine, and it's a green process, only uses green solvents, so it goes in line with the sustainable idea that we want to have in our final product.
Okay, thank you, Catarina.